Hello guys, welcome to our final week or the final topic for our online class. In this video, we're going to discuss about cloning. So what is cloning? If you have seen many movies, so what is cloning? Cloning is an organism that is genetically identical to its parents. So meaning it's just a copy to which it was taken. And this means that it has the same gene as its parents and will therefore look identical. So in humans, identical twins are actually a natural cloning. So if you have a identical twin or you have friends that are identical twin, then they are actually clone of themselves. Bacteria reproduce by dividing themselves in half. Each of them contains identical genes, resulting in two identical organisms. So this is actually how bacteria works. So this is a single bacteria here, and it will just divide itself in half and multiply. So as you can see on this video clip, so it will multiply until it goes to thousands and millions. Asexual reproduction. It is in bacteria, you do not reproduce using a partner, so it doesn't need any like male or female like humans do. So humans, for them to reproduce, needs to have men and women. And there is no mixing of genetic material, so the offspring produced are clones. So it's exactly the same. It is exactly identical to the ones that being produced. Plants can also produce sexually. So for example, for plants, the potato plants, it produced many tubers, each of which can grow into a new plant. So I'll show you the picture later. And strawberry plants produces long stems with tiny plants or what we call plantlets on the end. And the daffodils, at the end of each growing season, a new bulb is formed. So here, this is the picture for strawberry plant. So this is actually the long stems here. So it will just extend its stem and produces a new strawberry plants, as you can see here. Like this is the plantlets, and these plantlets are also called runners. And also for the potato plant, so as you can see in the roots grew a new potato plant. And as you have seen earlier, the bacteria. So how can you make plant clones? So first, cutting a small section of the plant, allowing to grow into a new plant. Second, cuttings are dipped in a special chemical. So I have prepared a video for you to see. So there is actually, after cutting the plant, you put it in a special chemical called rooting powder to encourage the cutting to grow roots. And just play it.
Find a growth tip. This is a point from where the plant actively grows. Now generally your cuttings will be more vigorous if they're taken from a plant in a vegetative state that's producing stems and leaves and not in flower. Slice around 4 inches below the growth tip. Some growers will insist you cut an angle of 45 degrees like we're doing here, but this isn't a golden rule. Just make sure it's a clean cut and try not to bruise or crush the stem. After you've taken your cutting, remove any lower leaves or branches so that you have at least an inch or so of bare stem to insert into your rooting media. Once again, remember to work fast. Dip the base of your cutting into rooting gel or powder and insert it into your rooting media before placing it inside a propagator. And that's it. See? Told you it was easy.
Okay, so that's actually how you clone plants. So it's very easy if you have the materials, especially the growing chemical. Advantage of plant cloning. New plants are produced quickly. The technique is very cheap and it's easy. All plants are genetically identical, so they will have the desired characteristics. Okay, it's actually the same with selective breeding. So if you have the desired characteristics for your crops, then you will choose selective breeding. Disadvantage of plant cloning: reduces the variety of genes available. So there would be no other genes for the specific types of plants. So you cannot choose from the different species of plants. Increase the risk of disease for plants and a change in plant environment destroying a species. So you clone the animal, so you clone the plant, you cut it, and then you place it to not its natural habitat. It will destroy its habitat, so where it actually grows. So how can you make animal clones? Animal clones is actually the same with human clones. People don't encourage having a human clones because there is actually a moral issue in this. They said if you clone yourself, it would be like so weird that you might actually want to kill yourself because you would feel, according to movies, you would feel like it is actually you that you are looking at. So that's actually how weird it is. But there is for sure many people have been looking forward to having a human clones, especially those people who want to live for eternity. So these are the procedures in making clones. So it is actually applicable also to humans. So cell cloning, scientists clone human cells in the laboratory and use them for research into disease. So this is actually like the advantage of having a cell cloning or human cloning or animal cloning. It it actually makes them the lab rats, like test. But as you can see, there is actually a moral issue here. So it's like you're making a human being, but you're still killing it for tests. And another type of making clones for animals, tissue cultures. So new skins and cartilage can be grown into sterile environment. So this technique is used to grow new skin for burns and victims. So this second procedure might be good for people that needs help, especially those have burns or some defects in their bodies. So looking at this rat, it has ear at his back. So this ear can actually or can maybe help in people that have hearing problems. So the very first animal or mammal to be cloned is Dolly the Sheep using cell from adult sheep. So the first procedure that I have showed you. So uh, cell cloning and then the tissue thing. And she was cloned at year 1997. And here's how Dolly the Sheep was being cloned.
Welcome to Moo Moo Math and Science. In this video, I'd like to talk about animal cloning, how they clone Dolly the sheep. Basically, in order to clone another animal, you take a cell from one, then you take an egg without a nucleus, and then basically you get an offspring. But it's a little bit more involved. Dolly was the first animal that was cloned, and Dolly was cloned almost 20 years ago. Here's what happened. Step 1 was to remove a cell from the udder of sheep number 1. They then took this cell back to the laboratory and starved it in order to force it into an almost embryonic state. Next, they took an egg from another sheep, we'll call it sheep 2, and removed the nucleus. Step 3, they used electric shock and fused these two cells together. Step 4, they implanted this fused cell into a female sheep. They actually fused more than 200 cells, and one developed into a full adult. Dolly had the same cells as the original udder cells. What did we learn from cloning Dolly? The main thing was that it helped advance stem cell research, and it proved that the nucleus of an adult cell had all the DNA to give rise to another animal. Dolly lived for 6 more years and then lived from a lung disease. Anyway, that's Cloning 101. Thanks for watching, and Moo Moo Math uploads a new math and science video every day. Please subscribe and share.
They will take an egg from a woman and remove its nucleus, the part which contains her DNA. They will be left with a hollow egg. Next, they will take a cell from the body of the adult who is to be cloned. It's from this cell that they will make a new human being, an identical genetic copy. Once they have selected the cell, scientists will take its DNA and insert it into the empty egg. Then they will have to stimulate the egg to begin to grow. They might use an electric shock and add chemicals, the methods used in animals. No one understands how or why, but this process should switch the adult cell back to become the very first cell of life. The new embryo will be grown in a dish for a few days, then implanted into a woman's womb, where it might grow into a human being. This human would be a clone.
So here's another video, like how the cell is being produced. It's like how to remove the nucleus from the egg. So this is actually the egg, and this is the nucleus. So they're actually removing this one and then replace it with the ones that you're going to clone.
This video is prepared by my colleagues Dieter Egli and Kevin Egan. And you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona. And you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out, and then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. So if we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you two, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like a Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again, a little hole, and the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of what's called somatic cell nuclear transfer.
Okay, so that's actually how they are doing the cell cloning. So that ends our topic for cloning. So I hope you enjoyed our video, and we'll see you soon.